Okay, good evening. Just confirm me that am I audible to you? Um, just confirm me. Am I audible? Whether the PPT is visible or not? Everything is perfect, right? Let's wait for 1 minute. Let others also join. I'll start as today's class. Okay, fine. Good. I think everything is perfect. So without any delay, we'll jump into our syllabus of today's class. See, today we are going to discuss this fifth subtopic, that is chromosomal aberration and autosomal abnormalities. All right. See, we have a different types of aberration. But before that, in last class, I have discussed about the mutations. What are all the types of mutation? And we have completed the first thing, that is gene mutation completely. And the remaining pending will be the genomic mutations and chromosomal aberrations, right? And what are all the types of aberration means? There are different types, aneuploidy and polyploidy. What is aneuploidy here? Here, the number of chromosomes will be vary. That is what an aneuploidy, abnormal number of chromosomes. For example, Down syndrome, which has been, we are calling them as the trisomy at the 21st chromosome. And what will be the symptoms? What will be the disorders associated with this Down syndrome? Means intellectual disability, distant facial appearance. Okay. And how we can detect this particular syndrome is with the help of the karyotyping, FISH, and Q of PCR, right? And what will be the treatment? Supportive care and therapy will be the treatment. With respect to this polyploidy, there will be a more than two sets of chromosomes will be there in case of polyploidy, that is triploidy. And it is associated with a lot of miscarriage. And how we can detect this particular disorder means with the help of the karyotyping and FISH. Okay, here the treatment is it is inapplicable. And we have a chromosomal aberration, that is deletion, duplication, inversion, and translocation. Deletion, we know that if a particular segment of a chromosome will be lost, that will be coming under a deletion. And the symptom associated with this particular deletion, that is one of the symptom or syndrome, will be the Cridle-Chat syndrome. And if a particular person is experiencing this Cridle-Chat syndrome, is what is the symptom experienced by that person means if he or she cry, that will be looking like a cry of the cat. Okay. And also an intellectual disability. And how we can detect this particular syndrome is karyotyping and microarray. And in duplication, if a particular segment of a chromosome will duplicate, then it will be coming under the duplication. And Charcot-Marie tooth disease will be associated, or it will be result as a result of the duplication, and it results in the muscle weakness and atrophy. Okay, and once again, karyotyping and microarray will be the detective methods. Inversion, we know that a reversal of a chromosomal segment. For example, see the inversion occurs in the 9th chromosome, in the short arm at the 12th place, and in the long arm in the 13th segment. Okay, this is what an inversion example. And here there will be no effect as a result of this particular inversion. And this karyotyping and FISH, this will be the diagnostic methods. And as a result of translocation, if a particular segment of a chromosome has been rearranged between the non-homologous chromosome, that is what a translocation is. And what will be the disorder associated with the translocation means chronic myelogenous leukemia. Okay. And once again, a person will experience a leukemia. It's somewhat related to the blood cancer, that is WBC, abnormal count of WBC. And once again, we can detect this particular disorder with the karyotyping fish, also a polymerase chain reaction. And we have a different types of ploidy. First one is haploid. We know that a single end, that is only one set of chromosomes. An example will be the sperm and the egg cells. We know that sperm is haploid, egg is also haploid. As a result of a fusion, as a result of fertilization, it forms as a zygote, that is a diploid. Okay, and initially this germ cells will be in a haploid position, haploid in number, and that is what karyotyping and sequencing will be the detective methods. And we know what is a diploid. We have a two different set of chromosomes, and triploid means 3n. A tetraploid means 4n. That is what types of ploidies. In tetraploid, we have a four set of chromosomes. It will be appear in plants and also some species of amphibians. And how it looks like? Of course, it will be a 4n. It looks like a larger in size, right? And in triploid, it results in sterility and some of the developmental abnormalities. And this triploid will be detected in seedless watermelons and some of the banana variety. And what will be the type of polyploidy? A first type will be the autopolyploidy and allopolyploidy. Autopolyploidy means when multiple chromosome set originate from a single species. This results errors. This can be found as a result of the if we find any errors in mitosis and meiosis, this autopolyploidy will be occurred. Okay, what is that? A multiple chromosomes sets originate from a single species. That is what an autopolyploidy. In case of allopolyploidy, species. Okay, this is what an allopolyploidy. And we have a further different types of autopolyploidy, that is autotriploidy, autotetraploidy, and autohexaploidy. And also just aware of the examples. So that is an important thing. Autotriploidy, that is seedless watermelons and bananas, we can find there. And in autotetraploidy, we can find in the potato, alfalfa, right? And in autohexaploidy, we can see in the chrysanthemum. And in allotetraploidy, we can find in the wheat and cotton. And allohexaploidy, we can find in the bread and wheat. Okay, these are all the types of ploidy. Okay, now come back to the question. Which of the following is an autosomal abnormality? Oh, I forgot to add syndromes. Just answer for this question. Just give me 2 minutes. I'll show the list of syndrome, and after showing that, we'll solve the questions. Okay. Just 2 minutes. Wait. So everybody can see the picture, right? See the table. Just focus here. See Down syndrome. In Down syndrome, what will be the condition we can see is trisomy 21. Okay, trisomy 21. We can find 47 chromosomes because here we can look like this. 2n plus 1 is the condition of trisomy, right? And we can find 46 chromosome. Generally, 46 will be the normal chromosome plus 1 is the we can find in the Down syndrome. So we can find 47 chromosome and double x or plus 21, or we can find 47 xy plus 21. This is the condition in case of a Down syndrome. What will be the characteristic features? Intellectual disability, characteristic facial features, and heart defects. These are all the symptoms experienced by a person who have Down syndrome. In Edwards syndrome, we have a trisomy 18. So these are all the things we can find a question from the match the following question. So it is an important thing. You have to read this. Edwards syndrome, trisomy 18. Okay, this is the condition. 47 plus XX. We can find in female as well as in male. This is the severe developmental delays, heart defects, and clenched fits. These are all the symptoms. These are all the characteristic features seen in the person who have Edwards syndrome. Patau syndrome have a trisomy 13. See, trisomy is also an important number of which chromosome. The chromosome, place of a chromosome is also an important thing. Okay, and intellectual disability once again, and cleft lip or palate polydactyly. This is the condition we can see in the Patau syndrome persons. And Turner syndrome, we can find here that is -1. That is the 46 will be our normal chromosome. Here one chromosome will be absent here, right? And that is why 45 chromosome, and here one of in the sex chromosome we can find one chromosome will be absent in the sex chromosome, right? And short stature, webbed neck, infertility because it has happened in the sex chromosome, right? Heart defects. These are all the things we can find in the Turner syndrome. Klinefelter syndrome. Once again, this will be the 47th chromosome. And in the sex chromosome, we can find one extra chromosome. So we can find male characteristic features, also a female characteristic features. Tall stature, infertility, gynecomastia, learning difficulties. Gynecomastia, these are related to a gender problem. Okay, because female is also there, male is also there. Okay, that is what a Klinefelter syndrome and triple X syndrome. And triple X syndrome means, see, once again, it results in the sex chromosome. Here, mild symptoms, possible learning difficulties, a tall stature. These are all the characteristic features in the triple X syndrome. In the XYY syndrome, we have a 46 chromosome. That once again, that will be a problem in the XY chromosome. Once again, same thing, a normal development. At last, the development is affected here. And Cru-du-Chat syndrome, as a result of a deletion, we can experience this Cru-du-Chat syndrome. And that will be happened in which chromosome? That is an important thing. And also Williams syndrome. Williams syndrome will be occurred in which chromosome? See, what will be the type of chromosomal aberration? That is, whether it is a deletion or duplication or some other chromosomal aberration. And also you have to focus on what will be in which chromosome this deletion is happening and in which arm. That is also an important thing. Okay, you have to focus all these syndromes. This is an important thing. Okay, now come back to the question. Now answer for this question. I hope everybody has answered. And autosomal dysabnormality. Autosomal dysabnormality will be obviously the Down syndrome because there will be an extra chromosome occur in the chromosome 21. Okay. Now, how is Down syndrome typically diagnosed prenatally? Prenatally means what? Before birth. How we can find whether this baby will be experiencing this Down syndrome or not? How we can find this? That is amniocentesis, because we can take the for diagnosis we should take that amniotic fluid for the analysis. Okay, by taking this fluid we can easily come to a conclusion that whether this baby is experiencing the Down syndrome or not. Okay, which of the following is a symptom of trisomy 18, that is Edwards syndrome? Webbed neck, cleft lip, clenched fist with overlapping fingers, gynecomastia. That will be a clenched fist with overlapping fingers. This is what a characteristic features of Edward syndrome. Kindly, you have to know what will be the whether it is a trisomy or nullisomy or monosomy. That is, you have to focus on that. Also, you have to focus on the characteristic features. Okay. What is the genetic basis of Turner syndrome? Extra X chromosome in males, missing or partially missing X chromosome in females, extra Y chromosome in males, missing or partially missing Y chromosome in males. What is the genetic basis of Turner syndrome? See, this table is very, very important. Okay, in this Turner syndrome, what a person is experiencing. What is that? Missing or partially missing X chromosome in females. Okay. How does autosomal triploidy differ from autosomal tetraploidy? Triploidy has 3 sets of chromosome, while tetraploidy has 4 sets. Triploidy has 4 sets and tetraploidy has 3. Triploidy and tetraploidy both have 3 sets, but tetraploidy have a involve chromosome sets from different species. There is no difference, both refer to the same condition. That's a simple question. That will be obviously triploidy means 3 sets of chromosome, tetraploidy means they have a 4 set of chromosome. What is the characteristic features of Down syndrome? Okay, what will be the answer for this question? That will be a flat face. Okay, Down syndrome is characterized by a flat face, small ears, and slanted eyes. Right? And what is the main health implication of trisomy 13? That is Patau syndrome. Heart defects, severe intellectual disability, polydactyly, that is extra fingers or toes, high mortality rate within the first year of life. Why this person is not answering the question? Okay, anyhow. High mortality rate within the first year of life. Okay, it has a high mortality rate within the first year of life due to a severe health complications. Right? And how does the autosomal duplication differ from autosomal deletion? Just a difference between deletion and duplication. Duplication involves the loss of chromosome, while deletion involves the gain of the chromosome. Duplication involves an extra copy of the chromosome, while deletion involves a loss of chromosome segment. Duplication and deletion both involves the loss of chromosome, but deletion leads to a severe intellectual disability. There is no difference. Obviously, duplication will be involved in the extra copy and deletion will involves in the loss of a chromosome segment. And which of the following is a symptom of trisomy 18, that is Edward syndrome? what will be the symptom of trisomy 18? That will be heart defects. Very good. It is associated with various congenital heart defects. What is the genetic basis of Klinefelter syndrome? Extra X chromosome in males, missing or partially missing X chromosome in females, extra Y chromosome in males, missing or partially missing Y chromosome in males. See, you always tend to confuse with the syndrome, Turner syndrome as well as the Klinefelter syndrome. So you have to make sure that you can know the difference between these two. In Turner syndrome, there will be a missing or partially missing X chromosome in females, but in Klinefelter syndrome, that will be an extra X chromosome in males, right? Okay. As this topic is very short, that's all for today. For tomorrow, you have to study and come the human genetics, human karyotyping, genetic disorders due to mutant genes, that is Huntington's chorea, inborn errors of metabolism, phenylketonuria, alkaptonuria, sickle cell anemia. With the also, see, you have to study this subunit. Also, all these we have a combined test for tomorrow, right? Genetics fully. Also, the last subunit question will also includes both entire genetics. All right. Polydactyly is a concept we have studied in which area? In the non-chordata and chordata. Polydactyly will be the condition we have studied with respect to which class under the chordata? You remember? Polydactyly means what? Extra fingers. Polydactyly will be an extra finger, right? Like this. See, generally we have 5 fingers. This is what a polydactyly. Okay, clear? Extra toe, whether it will be in a hand or it will be in a leg finger. Right? Are you clear? Yeah, for tomorrow, the entire genetics will be the test for us. And after finishing the genetics, we are going to see a system and cell physiology. Okay? Everybody clear? Shall we wind up the class? Okay, fine. Thank you. We'll meet tomorrow the same time at 8:30 p.m. sharp. Okay. Thank you, everyone. If you have any doubts, you can stay, or else you can leave the class. If you have no doubts, just leave the class.